This is the brief overview of the contents of my presentation. I will not go into much detail of this. So I will start with the journey of the establishment of my lab, the plant cell signaling lab in Pakistan. I started this lab in 2012 when I joined the University Multan with the mission to explore the cell signaling complexities during plant immunity and management that would be the climate factor like drought, high temperature, radiation, that could be due to the phenological stages, and that could be related due to the biotic factors due to viruses, bacteria, fungicide. Plants have developed strategies in which they have made certain morphological, physiological, biochemical modification and of immunity. The first one is known as, this PTI is not personal sub, it is pan-trigger immunity. And the other type is the effector trigger immunity. What is effector trigger immunity? In this case, in case of effector trigger immunity, if you could see this picture, the first one, PAMs are actually the molecular pattern that are secreted by the pathogen when they attack to the plant, or they can be plant related patterns, or they can be secreted by the pathogen. These are recognized by the pattern recognition receptor on the plasma. Some are very early responses that start within seconds of the perception of the signal or the perception of the when pathogen attack to the plant. Within second times, the signaling cascade started in the plant. And among those early signals, there is the fluxes of ions, calcium ions, production of reactive oxygen species, nitric oxide burst, activation of protein kinases, then phytohormone biosynthesis. Signaling. In the first line, level of calcium increases inside the cytosol, that is known as cytosolic calcium. This cytosolic calcium can come from two paths. Either it will move from the outside of the cell, or it will come from the cell compartment. There are certain of the calcium is could lead to 80 millimolar, and free calcium could lead to 0.25 millimolar. Apart from the vacuole, mitochondria, it moves outside of the cell. We say efflux of the calcium. So there are different transporter channels. You can see in this figure there are a number of channels. You can see here which are present on the double membrane structure or the plasma membrane that lead to the movement of the calcium. What we are focusing on these glutamate receptor-like channels that are present on the plasma membrane and lead to the movement of the calcium from such genes are present in the plant system or not. So in 1998, a group of researchers working in France, they observed that there are 20 glutamate receptor-like genes present. They have no central nervous system in plants. These glutamate receptors lead to the calcium homeostasis, calcium-related NADH oxidation, long-term program cell death, abscisic acid reduce calcium deactivation. Abscisic acid that is come in the downstream signaling pathway. We have selected three different pathogen Yellowpyrospora rhabdopsis, that is an OIGs. Then what condition for this plant? We have built a special culture room with very controlled condition, sophisticated infrastructure. So we can obtain these Multan line from Nottingham Stock Center, UK. And then we sow these plants in special soil that is known as peat moss. In the first step, we have to screen these mutant lines for the to check the homozygous status, because technologists, they know very well that allelic variation could also play the role in different role in the expression of the gene. So we selected 2-3 different mutant lines and genes that are mostly playing the role in the signaling process. So after this confirmation, we get another mutant lines of these 3.3 mutants, and you can see the dark graph in the first graph, that is of the peak of the wild type, and time, if you can see, it is in seconds. So we obtain the peak at around 30 to 40 seconds, and 30 seconds gene. This is the marker gene for reactive oxygen species production. We observe the expression of PAR4 gene. This is again a gene that is oxidase pathways. And we observe the expression for PR1 pathogenesis related one gene that is and we observe the change in the expression of different physiological and biochemical parameters. And we observe that treatment of the salicylic acid we also perform transcriptomic analysis with these glutamate receptors, and we find some key regulatory transcription factor, and we selected these two very transcription factor name as mutation based on the activation of glutamate receptors, and we said that glutamate receptor 3.3 is involved in this process. So we checked whether in the mutant line of these wiki, what is the expression level of glutamate receptor 3. When this calcium is accumulated, it leads to the production of reactive oxygen species, it can lead to the production of nitric oxide, it can lead to the activation of defense-related gene, and if we stop the entry of these calcium channel by their blocker, by their inhibitor, glutamate receptor genes transgenic route that can express these glutamate receptor overexpression, and that can be more resistant crops. So, I would like to thank our collaborative partner and the funding agency in Higher Education Commission, United Nations Office for the Outer Space Affairs, all the team members of